Hi students, in this video I am going to explain you about the structure of proteins. Because proteins already discussed and so many times I told you people that proteins are the basic bodybuilding units of the cell. And it is most important for the Neet also up to now only one bit here. But it is important for many aspects for the any exam. Now, if you see structure of such a type of the protein which is to occupy major organic compound in the cell, there it came, organic compound in the cell. It used to have so many type of the structures and do so many types of the functions. So it is very important in that aspect also. Now the proteins already last video only protein structure whenever we are discussing. Proteins are the heteropolymers made up of the monomers of 20 amino acids only. 20 amino acids used to form the one in different ratios it used to form one protein. And not only that one, it used to form different structures, and these structures are suitable for the doing of the different types of the functions. So proteins are the polymers, heteropolymers made up of the 20 amino acids. And now whenever we are seeing these whenever we are they are doing seeing the protein, generally the any type of the compound or any type of the arrangement whenever we are discussing in the inorganic chemistry, we used to say as the formula only it can explain the structure, and organic chemistry it will be the two dimensional and it will give some structure. In physics also it is three dimensional, but here in the proteins it four level structures is to present. That is the most important. Not only one type, it is the four levels of the structures can be observed by the same proteins, which is that protein primary protein used to make the other three types of the proteins. In the structural difference it is to present. Proteins are the strings of amino acids in heteropolymer form. It is a heteropolymer because different monomers used to present. Protein structure present in four levels. What are the four levels? Now you see here. That is the primary, secondary, and a tertiary, quaternary. Four types of the four structures it can make. But all these three, secondary, tertiary, and quaternary, will be done by the primary only. Remember it. And in the whenever in the translation, whenever the protein is preparing, that will be the primary protein only. That primary protein will undergo different structural differences. It will go undergo the difference in the structural differences and forms the secondary, tertiary, and quaternary. So without primary protein, it cannot be. Other proteins cannot be formed. Primary protein is most important one, and primary protein only will be synthesized by the mRNA when it comes in the translation. These are the important point, remember it. And whenever the primary protein we are seeing, it is a string, means fiber-like structure. It used to have one all the amino acids used to attach one to another in particular manner, and they are bonded by the peptide bonds. This is the peptide bonds which is present in between, and we can observe the primary as a string, and first amino acid, second amino acid, third amino acid, how it is we can say. At the same time, he has given one more thing also, left end and right end. The left end, whatever is there in the protein, first amino acid which is present in the left hand, that protein, left hand of the protein, primary protein, is called as N terminal. What is N? Amine. NH2 amine. So amine terminal, N terminal, they are calling it as a amine terminal, they are calling it as N terminal. And right hand side, whatever is there, that protein right hand is called as, it is the C end, carboxyl end. That is nothing, carboxyl group. Now this C yours in the 23 means right hand is the, he given it as a wrong statement, and left hand is the right hand, one statement, and second statement is correctly has given. So only one question came from this. Now the thing is, these primary protein actually it won't do any function. Only it will form the different kinds of the proteins, different levels of the proteins. Means physiological function is it won't do anything. But it by modifying structural modifications, it used to do many functions. What are there? Now first we will see primary string-like structure. Functionally it is a 0. But it can do function, all the functions by structural modification. Remember it. These are not given in the book, but the thing is, if you know this concept, then only you can give the answer. Now, how they will do? They will form the secondary. What is secondary? That whatever the string is there, it used to modify. Yeah, the spiral. That is helix, like formation, like this. That also right-handed helix. That also he has given right or left. This also important for the Neet. So right-handed helix it is to form. And beta plated sheet. What is the beta plated sheet? Means here it is nothing. The same thing used to form long loops like this. One of the part whenever we are seeing this, these are the beta helix. Means long loops it used to take. It is spiral, this is helical. And it is a long loops like this it used to take. Whenever we are taking this one, it used to have. And bonds also difference is there. Here alpha helix bonds will be taken whenever the spiral is taking place. Now you see, whenever it is bending, then only the bonds will be there. In between there won't be any bond. But here, whenever we are seeing, whenever the long loops like this they are taking, in between the two parallel strings of the same protein, bonds will be taken. And in the secondary protein, only hydrogen bonds will be there. Hydrogen bonds only are cause for the formation of the beta plated and alpha helix. Now, the thing is tertiary protein. And this he has given like this is arrows. You are observing, no? Means it may form this one or may form this one. Now both the things he has shown this one. Why? Why anyone, very few people used to explain it. The thing is, here you see, secondary and beta plated both are present in a single primary protein. Very lengthy primary protein, a portion, a particular portion will form the alpha helix and particular some portion will work as the beta helix. Means some will be like this, later on it will be like this, then it will be hollow woolen, woolen ball. That means like this, means like this spiral. Then immediately it will become this whatever the beta helix. Immediately it will form the this thing. Why it is forming like this? That is also very, very important. Now you see here. Here he has shown clearly. Whenever we are seeing, here you see. This he has shown helical and next beta plated and next all this rounded. This is self-coiled hollow woolen ball. He is telling. in the self-coiling of the primary protein, and here and there in some portions they used to form the alpha helix and the beta plated. All together will form the tertiary structure. You will get the doubt why it has to do. Now it has to participate in many of the functions, many physiological functions. What are they? Now you see here: primary, secondary, tertiary. So this is tertiary. Yeah, it is formed. Now I discussed already. Now you see here, the tertiary is the tertiary form. A portion become the alpha helix and a portion become the beta plated, and then our string will become self coil and tertiary structure will be formed. The tertiary structure will be there. It will be most majority of the physiological function will be done the tertiary structure only. And one more thing here you should remember, that is all the enzymes are nothing, only protein. Enzymes used to have the active sites. Enzymes are having the active sites. That it will come in the next video. The active sites will be formed by alpha helix and beta plated. For that only it is having. What active sites are formed by alpha helix and beta plated, which are taken within the tertiary structure. That is the cause. Now, this why he has given quaternary structure. Again, arrow he has given. This arrow has given because both are present in this. And it is mostly useful. The proteins are here, many biological functions done by tertiary structure. What are biological structure functions? Mainly enzymes and other structural functions also it will be. So it is to have that much facility by having the alpha helix and beta plated at some places. Now, whenever we are seeing once more, that is, here you see. Now, once more we will see here, that is tertiary. Tertiary here shown from the quaternary, from the tertiary structure. Here again we will observe one thing, that is, it is a complex proteins are present and doing same function is the quaternary. That he has not mentioned, but simply he told different types of the proteins used to act as the subunits and do the function, one function. And it is also most important quaternary structure. How it is? It may be the spheres may form, cubes may form, or some other secondary structures may come and form. For that one, he has given one example, that is, proteins become subunits and forms quaternary structure. In this one quaternary structure, it is not a single primary protein, but secondary, tertiary, or single protein only, single whatever the primary protein. But here it is to have the so many primary proteins which has modified or unmodified. So here the thing is, for example, he has given hemoglobin consists of the 4 subunits, means 4 primary proteins which is modified. 4 subunits, 4 types of proteins are present in the formation of the hemoglobin which is in the human body. Now 2 subunits in that one, 2 are one type and 2 are another type. What are they? They are the primary proteins which are formed the alpha helix, beta plated. Alpha helix 2 proteins and beta plated 2 proteins. 4 will become like this. They will be one upon, but this is the beta plated, and this one, and like this like this. They used to become compound protein. We can say it compound protein also. because number of simple proteins are present. But proteins are simple proteins means not primary. They converted in the different tertiary proteins also used to participate. Different tertiary proteins used to participate in the formation of the quaternary. Also remember it, because tertiary and quaternary proteins are most important for the functionality. But tertiary protein can be formed with one protein. In that protein, the primary protein alpha helix and beta plated is formed at different places of the same protein. And finally, string is formed as hollow, self-coiled hollow woolen ball. That is. But here, it is a different types of the protein. Proteins number will be different, shape will be different, and finally it will become complex protein and it will do the one work. For example, he has given the hemoglobin. I think you might have understand the properly. And note the points, important points, because he didn't touch this one. In any which angle he will touch in the Neet, we don't know. But it is one of the most important. If you like this video, please give the like mark and subscribe the channel to see all my videos. Thank you.